Good afternoon. Welcome to our seminar today that's taking place both here at the LMB and online as part of Cambridge Biomedical Campus virtual tours. So as always, just before we begin, a few technical details. At the end of the talk, there'll be an opportunity to ask questions. I really encourage you to do so. If you're participating online, if you could write those questions in the Q&A, then I will read those out for you. So just a few words about the LMB. So the MRC Laboratory of Molecular Biology, or LMB, is a world-leading research institute situated on the Addenbrooke's Biomedical Campus in Cambridge. Our aim is to understand complex biological problems at all levels, from atoms and molecules to cells and organisms. As a centrally funded institute, our remit is to study difficult long-term problems, and in doing so, contribute knowledge needed to solve key issues in human health and understanding. Over the years, LMB scientists have made many contributions, both in terms of new understandings and in the development of new technologies. Although we are a molecular biology institute, our research covers many areas, and today I'm delighted to welcome Sam Roberts, a chemist from the PNAC division, to give our talk. Sam is a postdoc in Professor John Sutherland's group at the LMB. After an undergraduate master's in chemistry at the University of Oxford, he tells me he was unable to escape the southeast, moving instead to London to study a PhD in chemistry, again on the origins of life at UCL in Professor Matt Powner's lab. Sam then moved to Cambridge, briefly working away from the origins of life in Professor Shashanka Balasubramanian's group, where he worked on G quadruplexes in DNA, before starting in the Sutherland lab in 2021. He holds a junior research fellowship at Wolfson College in Cambridge, and today he will tell us all about the chemical origins of life on Earth.
Thank you very much for everybody coming along and for inviting me. And yes, so today I'm going to be talking about, or trying to provide some reasons why you've got a load of chemists kind of infiltrating your biology department here. Origins of life, I think, is kind of an inherently interesting question. It's something the question of where do we come from is one of our oldest questions, and seems to be something very particular to humanity that kind of wants to understand its roots. But unfortunately, we don't have time, or nobody's yet invented a time machine, so we can go back and actually find that question out for sure. So what we are trying to do in our lab and various other labs is try to work out how life could have arisen on other planets, and as well as kind of trying to understand our potential origins, it's also starting to inform how we go looking for life elsewhere in the universe. So we have this kind of these two very different pictures of life on Earth. We know what or we have an idea of how the Earth might have developed. You know, it's a very rocky, desolate, harsh environment with kind of very simple molecules. Think things like CO2, nitrogen, and so on that we're familiar with in the atmosphere, and various minerals and so on. But definitely abiotic. There's no life present, and thanks to biology and institutes like this one, we have an understanding of what life looks like now. We have a vast variety from everything that's kind of things that we can see, like ladybirds, and all the way through to tiny microscopic organisms, like bacteria and so on. But there's this big yawning gap about how we transition from one to the other. And we feel like we have a good definition, or we have an idea of what is alive and what is not, but there must have been a transition between those two things. So essentially what John's lab is trying to work on is essentially, what does this arrow mean? What is this process going from A to B? And the reason that we feel it has to be chemists who asks this is because before you have, so biology is life. So before you have biology, you have pre-biology. So this is what we call the prebiotic world. And the interactions of biology are governed entirely by chemistry. If you look at the individual parts of biology, they are governed entirely by chemical interactions. And they are themselves chemicals. So to understand where these chemicals come from and where the processes of biology come from, you have to bring it back down to chemistry. And so that's why we feel we're in the best position of being chemists, but in a biological institute such as this one, to kind of bring those two ideas together. before we kind of move on to how we start to solve this problem, I thought it's worth kind of having a frame of reference of kind of scale, which is kind of useful as an idea of like how complex some of the transition might be. So if you imagine the very first forms of life, we blow them up to the size of this room, this lecture theatre. The molecules that would have been available on the early Earth to start building biology would have been about the size of a football. So we're scaling up several orders of magnitude to go from a football size to the first microorganisms, which are about the size of bacteria, scaled up to the size of this room. And for reference, we would be about the size of the width of Wales at that kind of size. The way that I like to think about how we go about this arrow is a bit like how you might build a puzzle. So when we know everything, you'd have the completed picture, or when you produce biology, you have the completed picture of the puzzle. But you don't necessarily build the puzzle in like a particular order going left to right. You find the bit, you solve the bits that are easiest and more unobvious to put together. So for instance, corners and edges, right, are often easy, or more distinct parts of the image, such as the polar bear and so on in this particular one. So whilst we haven't necessarily we haven't got the complete picture all put together, we are taking puzzle pieces and seeing how they fit together and trying to construct a coherent image from that. And I would also say that it's not just like we've got one possible puzzle in front of us. We've basically got thousands of different puzzles all in a giant box, and we're trying to fish through all the pieces and seeing which ones fit with which. So let's get on with some of the science. So the earliest experiment in this kind of field, and one that is kind of seems to be a little bit in the public consciousness, which you may have heard of, is called the Miller-Urey experiment. And basically what Stanley Miller tried to do was recreate the conditions of the early Earth. So let's see if I can, does this work? So in here we have, I'm getting used to this, there we go. In here is essentially a container with a load of the conditions that they think, that at the time they thought was reasonable for the early Earth, for the origins of life essentially, and which they called the prebiotic soup, hence the primordial soup. And then they threw a load of spark discharge, electrical spark discharge through here to simulate lightning, and then have a look to see what comes out of here, and kind of try and make assessments about how that could have led to early life. So to us, this is kind of not particularly scientifically useful experiment nowadays, because this is akin to basically taking all the puzzle pieces possible into a box, shaking them, and then looking and seeing if we've got a completed image inside the box. Okay, so even if this experiment had worked and had produced life or something that's on the way to life, we wouldn't know how it had happened. We just know that it had happened. And it didn't produce, and even then, even if you think that's interesting, it didn't produce anything. In my opinion, it didn't produce anything that interesting. The most interesting thing that this experiment provided was it was the first experiment that took something that was definitely abiotic and then tried to get it towards biotic. So taking something that's purely chemical and tried to see how close to biology can you get to, which was a paradigm shift at the time. So we don't like this kind of black box shaking the puzzle box type approach. So how do we go about this in a more scientifically satisfying manner? So let's start. So we can look at the start point at the end point. So the start point is our surface that we're going to build our puzzle on, our parents' living room floor or wherever it is. This is akin to the early Earth, before any kind of biology has appeared. And we can start to put some constraints on the chemistry that could have occurred on early Earth. So the first thing is a timeline. So we can follow life all the way back to roughly 3.5 billion years ago. So that's 3,500 million years ago, the fossil record goes back to. So we know that life must have existed before that point. We also know the Earth itself is 4.5 billion years old, but it's generally regarded the first 500 million years or so is too violent a period in the Earth's history for Earth to have formed, or if it did form, it didn't survive. We've got lots of meteor impacts, lots of volcanic activity, and so on. So generally not considered. So it's generally not considered kind of relevant for us. But it does mean that we have roughly 500 million years to work with, which means my 3-year PhD was kind of a very small portion of that, and it means you have to think quite carefully about the experiments you run. And we can also make some assessments about the chemistry that would have been available. Much like the modern day, there would have been lots of water about, but some debate about exactly how much water in condensed in condensed oceans and lakes and so on. But the actual chemical would have been present for sure. And we can also kind of rule out certain things. There's definitely no chemists in white lab coats, kind of carefully mixing things together in certain orders. So that is important in how we think about how the chemistry must have happened. And also certain chemicals that maybe we take for granted today wouldn't have been around. So oxygen is the classic one, because we actually think that oxygen appeared a couple of billion years later than this, and was actually a waste product of some of the early forms of life. So we have to think about that in terms of the chemistry, because there's oxygen in the atmosphere now. So when we think about the chemistry that we do, does the oxygen in the atmosphere essentially affect that? There's no oxygen, so there's also no ozone, because ozone comes from oxygen in the modern atmosphere. But there is still a star, our sun, which produces lots of UV light. It would be a bit different because it's it's quite a long time ago, so the way it emits light would be different to now. But there would be quite significant UV radiation that would have hit the surface of the early Earth. And we know that UV radiation can do things because it does. It interacts, for instance, with DNA to cause cancer and so on. So so the UV light is bringing energy down to the surface of the Earth that can be used in chemical transformations. And then there's all the kind of other things that we almost kind of take for granted now, that you'd have volcanic eruptions, so which would include various gases that would come from that, for instance, sulfides, lots of minerals that can do interesting things, and we also have atmospheric chemistry, which will become relevant for later on. But beyond some of the hard constraints like oxygen, and the fact that there's water around, we don't try to limit ourselves with possibilities too much. It's more about what kind of plausible environments could have existed. And I think one of the kind of key things is that, much like the modern day, the specific environments are going to change dramatically from place to place. So the surface of a volcano is completely different to the deep ocean, for instance. So what about the other end? From our complete puzzle, what does kind of current life look like, and how can we trace that back to start to understand where it comes from? We have all these. There's 3 different main families of life, and we're in the eukaryota, up in animals, up here. But you're familiar with bacteria, and archaea are kind of to me, because they're usually single-celled organisms, they're kind of closer to bacteria, but I'm sure people who think about this more would disagree with me, because they're one of the oldest forms of life, for sure. But all 3 of these branches of the tree of life converge to a single universal common ancestor. So if you go back far enough, all of these, even though that we're vastly different in our biology today, have a common ancestor. That doesn't mean to say that there weren't other branches of the tree of life that have since died out and that we don't have any evidence for, but we can follow back the current forms of life to this last universal common ancestor, Luca. So any traits that would be found across all the different forms of life are likely to be present also in Luca, because evolution of traits or biological processes, it would be very unlikely. It's very unlikely for things to happen kind of multiple times, essentially. It is known to happen. So, for instance, for some reason, crabs are a really good template for life, and I think crabs have evolved something like 3 or 4 times on Earth as it's kind of over its lifespan. But in general, you can look at the common features of all the different forms of life and say, okay, it's likely that if they're common, that they are ancient. One of the kind of trait or feature that we're most interested in is these three molecules here, or these three species here. This is known as the central dogma of molecular biology, coined by Francis Crick, who used to be lately of this parish, Nobel Prize winner. And essentially, what this diagram describes is the interaction of these three molecules and how they're useful for life. So I'm sure you're familiar with DNA and proteins, but maybe not RNA so much. So the way that I think of this relationship is to compare it to computers. So if DNA is the ones and zeros on a hard drive, and proteins are your outputs, so they're your, the printouts from your printer, the sound from your speakers, and so on. RNA is essentially the software that's going from one to the other. So it's translating the information of DNA into the information of proteins. Much like that analogy, and the analogy is very simple and the diagram is very simple, but that kind of hides quite how complex both those things are. And in reality, this is just very, very complex, and it's hard to see how this could have appeared spontaneously as it is now. So the question is, what preceded this organization in biology? And usually that question is reduced down to which of these three things came first. And usually the one that's won out the most over the years is this molecule in the middle, RNA. The reason for that is that, as well as its kind of modern software type role, RNA can also act as a data store. So for instance, in viruses, we're all familiar with COVID-19, that's its genome, its genetic information was actually stored in RNA rather than DNA. And RNA can also act as a functional molecule. It can do things. So, for instance, the ribosome, which is a molecular machine made up of RNA, is the machine in biology that's across all biology that produces proteins. So this has led various people to suggest that, in fact, the first form of life looked like this instead. RNA acting as all three, having all three roles in the first forms of biology. And this it was kind of encapsulated in a title called The RNA World, which maybe you have heard of a little bit. Now, this is RNA. Organic chemists start to wake up and kind of get excited because this is actually a chemical now, not just pretty pictures. But it doesn't take an organic chemist to kind of say that this is a really complex molecule, and that it's kind of it's very hard to see how this molecule could have developed. It's not a trivial problem to understand where this came from. So this has meant that the origins, or at least a large portion of the origins field, has now kind of seen the RNA as kind of an important stepping stone, an important checkpoint on the way to life. And that the problem, instead of just being kind of an arrow, straight line from the early Earth to life, is now how do we get from the kind of rocky planet that's completely devoid of life to one that has RNA on it? And then from RNA, how do we get to something that we can actually define as life? I will give a shout out to Phil Holliger's lab here, also at the LMB, who are interested in this second arrow in particular. We're also starting to make moves in this direction as well. So they're interested in replication. So replication and evolution is probably one of the key defining characteristics of life. And they're trying to understand how replication could have got started from molecules like RNA. We're interested in how RNA and the relationship between RNA and proteins would have first appeared. But what John has mostly focused on is this first question about how does RNA appear. thanks to partly because John has spent so long working on this, and also various other researchers over the years, we're now starting to make the puzzle actually look something like this, partially kind of complete, where there are definitely still gaps and holes and things that we still need to find out, but at least for our scenario, we're starting to see, the image as we'd hope it would be kind of come out of it. However, the second step, understanding what RNA can do and how we get to life, is a lot more at the kind of jumble of pieces in a box stage at the moment. So what I want to spend the last few minutes talking about is how we think this molecule RNA could have appeared, and essentially what we as chemists have been doing in the lab. So this, as I mentioned, is an RNA molecule. However, it doesn't actually look like this, right? This is just a drawing to help chemists understand how RNA, or any chemical for that matter, actually works. What it actually looks like, if you took a photo of the molecule of RNA, it would look something like this. Well, this is actually two RNA molecules, which is where the two colors come from. But this is literally a photo, it's a crystal structure. So it's basically taking a photo of a crystal but using X-rays rather than light to see what the structure looks like. So this image is kind of what it actually looks like to us, but isn't useful because it doesn't tell us about how, or isn't useful for our purposes because it doesn't tell us how it works very much. So with that in mind, what I want to do is explain how we think this RNA molecule could have appeared. But we don't need all the information that's in this diagram for me to explain this. So I'm going to use blobs to simplify matters, but it still give you the information about how we get from the rocky planet to this molecule, okay? So there's a lot of stuff's going to come up on the screen, but I'll just walk you through it. What we think is we start off at the top right up here. Come on. No. No. Okay, this is the one bit where I actually need a pointer, so I will do it on here. No, that's not what I asked for. Laser pointer. Okay, red dot up there. So this is where our starting point. We've got our rocky planet. There's lots of violent things going on. So various atmospheric chemistries with lightning going on, but also meteorite impacts, which are also pretty violent, right? Think like a big rock coming out of space, generating a lot of heat, pressure changes, and so on. And both these things can do all sorts of exciting chemistry to compounds in the atmosphere and make this compound hydrogen cyanide. Hydrogen cyanide, I'm sure you're all aware, immensely poisonous and toxic to modern life. And so you may think, oh, that's not great. Why is this getting towards biology? But for us, what excites us about us is that the carbon and the nitrogen is very similar to all these carbons and nitrogens that we see in the bases here, in our RNA molecule. And this is what we have to set up to be able to build this prebiotically. And just because biological molecules might have first appeared on Earth in a way that's not in a way that's maybe toxic to life, doesn't preclude it from being the way that life initially got started, and then it developed other processes to kind of maintain itself. So with these kind of quite aggressive things, hydrogen cyanide could form in the atmosphere and then rain down onto the surface of the Earth and collect in something akin to the prebiotic soup that Stanley Miller used way back at the beginning of this presentation. By combining this molecule, hydrogen cyanide, in a series of reactions that would likely happen in this pool, and shining both UV light and bubbling hydrogen sulfide, which would come from volcanic outgassing. So think like the geysers at Yellowstone, not like a massive Krakatoa type event, but kind of relatively small streams of gases and stuff coming from the Earth. We can now put together the blue and the red blobs here, which are akin to the sugar unit here and this base here. And then if this pool was then to dry out and the minerals underneath the pool were allowed to react with it, we then can start to add in this star unit here, which is the phosphate group here. And continuous drying then starts to staple these units together because we have this repeating kind of motif, essentially, of the three blobs kind of all combining together into this one molecule. So essentially, big atmospheric chemistry, rain producing a pool, followed by UV light and in the presence of geysers, and then a period of drying allows us to build RNA under conditions that we would consider plausible to exist on the early Earth. So I should point out that there are other groups who think otherwise about how this could have happened. One of the things that comes up a lot in the kind of public sphere, so if you read articles on BBC News and things, is various people like the idea of hydrothermal vents being a source for life, which I'm sure you maybe have been aware of. But we think our scenario has the most evidence for it actually working, and is very different to a lot of these other environments. And like I said, there are still questions to be answered in this. I've kind of given you a very brief overview of how this works, but we think that the puzzle's getting close to being complete. And this is how we do it. So our lab on the second floor is everything's it's still organic chemistry. It's still using round bottom flasks and lab coats and so on in fume hoods, but this is how you have to ask. This is how we feel you have to actually ask the question to actually understand the individual processes. Actually look at the individual puzzle pieces, not just try and magically kind of shake the box and hope that you statistically get life out at the end of it. So yeah, thank you very much for listening. Thank you to all my colleagues who are great people to work for.